Welcome. This is part 2 of the FlowCytoScript tutorial. In this part, we're going to talk about how you are going to take your flow files and prepare them for use in the FlowCytoScript. For this, you're going to be using FlowJo. If you don't have a copy of FlowJo, go out and get a dongle or talk to somebody about getting a copy of the Slight license now. Right, so for this, the first thing we're going to do is locate our data files that we want to use. If you're working with Aurora data, which most of you in the Babraham Liston lab will be doing, you're going to get something from the machine that looks like this, which is a zipped file. We need first to unzip this. So for this, if you're working in Windows, you go here and say extract all, and it might take a minute, and then you should get a pop-up asking you to unextract it. Once that's done, you'll get a version like this, and inside you'll have these folders, a raw folder, an unmixed folder, and some other ones for the experiment. The unmixed is what you want with Aurora data. This is comparable to what you would get for compensated data. The fluorophores have been unmixed into specific channels, one that corresponds to each fluorophore, or in our case, antibody signal. So only work with the unmixed data. Inside this unmixed folder, so this is the extraction folder from earlier. So we would hit extract and it would copy that. We're not going to do that right now. All right, so we already have these unzipped data. We're going to look at the unmixed one. And then here you can see that I'm working with tissues. So I have my group set up such that I have one group per tissue source, in this case blood, and then inside this you should have the individual FCS files that you're working with. And you can see that I have named these in accordance with the instructions on how to name your flow files, which you should review before running your flow experiment. And that includes the mouse name here, the ALFT, the mouse's genotype or condition, and in this case, a tissue source. If you have other variables in your experiment, those should also be in the name of your FCS file, which is going to help you sort the files later in the flow script. You also want an individual number attached to each file, so each file is distinct, and you can put them in order. So we're going to import these files into FlowJo so that we can process them for use with the script. So just select the ones you want. And drag and drop them into FlowJo. In this case, we're going to take samples from the blood, and we're also going to take samples here from the IEL, which is the gut. Now, as it happens, I already have an analysis worksheet for this one, so I'm going to open that up. And this is what a lot of you would probably be using if you don't know exactly what you're doing. You will probably have a template with a gating structure in it that you can use. This will be helpful for you to get to the populations that you're interested in analyzing with the flow script. So one thing we want to think about very clearly with the flow script is, what are we trying to analyze? What's the purpose of the experiment? That we need to figure out before we go to the script. So in my case, I'm looking at mainly regulatory T cells. So I need to isolate the regulatory T cells from each flow file and export those cells for use with the flow script. If you are looking at, say, a disease model and you're interested in knowing basically what's happening and you're using a very broad flow panel, you might just focus on live cells or live CD45 positive cells. If you are working on a CD8 project, you might isolate the CD8s. And the way we're going to do that is with gating. And you can, of course, do this for multiple populations. So what we've done here is we have files imported into FlowJo, and we're going to now restrict the analysis to specific cell types, such that the FlowScript can analyze those. So the first thing on the Aurora is that FlowJo doesn't work particularly well with the data, so it comes up automatically with side scatter height and forward scatter height. You want to change these to area, the A. And then we have a gate. In this case, I've drawn a lymphocyte gate just around the lymphocytes. You can do this a little bit more appropriately with a polygon gate that you set yourself. And then you can adjust the corners as such. You can also rename this, etc. By clicking on that gate that you've just created, you isolate only those cells that you have selected that are inside the gate. From there, the next gate is typically a singlet gate. So for this, we use the difference between the area and the height parameters. So we can click on this and change this to height. Now we see that the bulk of the cells are in one population here, and there's a second population here, and then there's a bit of a smear over here. This is the singlet, or so single cells. These are two cells stuck together, and then these are multiple cells stuck together. We want just single cells. We don't want to be looking at two cells stuck together. That will give us false signals. So you restrict it in that way. And then you can do the same thing for side scatter to be doubly certain that you're looking at single cells. And you can see here that basically almost all of those doublets are already disappeared. Whereas if we look at the entire population and look at side scatter and height, you can see that that doublet population is here. All right, so now we've looked at our single cells. And we can do that here. And then we'll gate on viable cells. So for this, you have a viability marker in your panel, and you can put that versus side scatter, or if you have a CD45 in your panel, you can already put CD45 on the axis and gate like that. So for this, you can draw a gate just like this. You should already have one if you're using a template. And the cells that do not pick up the viability stain are the viable ones. From there, we will move into the populations that we're interested in. So we might be interested in B cells with CD19, or we might be interested in, say, T cells with CD3. And then here, we can also change the way the data are displayed. And this is going to be important for exporting your data. So to do this, we change the transform, which is clicking on the T. We say customize axis. And here you can change on this slider bar for the width, how much the bi-exponential display compresses the negative. So you can see that with no compression, you actually have a lot of events that are on the axis here that are off scale. By compressing this, we can bring these together. And then if we hit save and apply, we can update that. And so you can see now that the negative is much less spread out and allows us to focus more on the positive events in the region without worrying about the noise and the negative. The other thing you can do is change the display on the maxima on the axis. So typically when you first open Aurora data in FlowJo, they'll be displayed as maxed out either at 5 by 10 to the 3 times 10 to the 6 or at 10 to the 5, depending on your computer. The correct is probably 5 times 10 to the 6. So if you have the data at something like this, you can see that some of the events are off the axis. This will create errors later in R, as some of the data will be effectively displayed as a max value when there is actually a range of values there. So I'm not going to do that. If your data are displayed like this, then just click on the plus button a couple times to get the whole range of data displayed. Right, so here we can say, I want my CD3 cells. We say CD3 positive, CD19 negative. You can drag that away and then we can adjust the size of our gates a bit here. And then once we have a gating structure, let's say we just want to look at CD3 T cells, we can drag and drop that up here on all samples. Then you see that this gate net structure now appears in the other ones. And then you want to go through your samples and make sure that this gating works for all the samples. You can see here that there's actually cells falling outside the gate because of variations in how the staining is occurring. So let's drag that over. And then once you make the change, you'll see that this has gone from being bold to being regular text. In order to update this to the other samples, we need to drag this up here into the same position it is in the gating structure above. So we drag it on viable for all samples at the top, and then we should be able to scroll through and see that the gate has been set correctly everywhere. And you can see here that we're now getting some negative cells in this gate. So you need to find a good compromise. So we might just move this a little bit up like that and then try again. Ideally, you wouldn't have this issue. All right, so now we have this. If there are other cell types, other markers in here that you can use to clean up your gate, for instance, if we had a macrophage marker or a monocyte marker, we could use that and then do further exclusion of cells that we don't want before proceeding, which would make it a cleaner analysis downstream. All right, so let's go with this. We'll set that like that. And then with this, what we could do is export the data. So now in order to export the data, we want to go back to that file structure that we created earlier. So right now I'm working in my data folder. I'm going to go to my analysis folder. And I have my CD3 T cells. Now, in this example analysis, I only have blood, pairs, patch, and spleen from 3 samples, all wild-type mice. I'm going to add more samples now that I have set up my gating. So I'm going to add the rest of my blood samples. I'm going to add the rest of my pairs patch samples and add the rest of my spleen samples. So now I have 24 samples because I have 3 groups of 8, so that makes sense. As you'll notice here, this matrix design is the compensation matrix. So some of these are blue and some of them are gray. By default, the Aurora data will come up with gray because there is no compensation matrix applied, or that's the acquisition defined default matrix. In some cases, you may need to apply a compensation correction, particularly if there's tandem breakdown. In this particular experiment, I had to do that. When we import new samples, the compensation is not automatically applied to all of those. So if I want to apply it to all samples, which is correct, then I need to go into the compensation. So tools, edit compensation matrix. We select the matrix that I have, the blue one, and then I drag this M onto all samples. Then we can close this. Now we are going to export these data. And to do that, we can just go up to this gating structure at the top and click on the population that we wish to export. If we wanted to export all viable cells, we could do that. So we could say, click here, right click, export concatenate group, and we want to export. So rather not concatenate, because that makes one file with everything. We want to export. And so in this box now, we're going to set a couple parameters that are important for us. One, you want to export all compensated parameters. No matter whether you have applied compensation or not, just always click that box. For events, always include all events. You can set this to limit it to a specific number of events. For instance, you're only probably going to analyze a couple thousand events or cells per data file. But by including all at this point, it allows you to later downsample in R and not have to go back to FlowJo. Now, open advanced options. You can see here we have a prefix called export. You don't need that. What you can do is put in other prefixes here. So for this particular example, we're not going to do that, but in the TSOL one that follows, I will show you how you can use this prefix or suffix to add tags to your data that allow you to sort it better in R. Finally, you need to adjust the destination. So right now, the destination will be where the FlowJo workspace is. We want to put it to have the files go into that analysis folder that we just created. So we just are going to export our viable cells. So I'll make a viable folder, select that. So you can now see that it's going to my data analysis folder, viable. And then with this set of parameters, we click export. If you click export while there is still a text down here in the lower left saying calculating, it will either take a long time or fail on some of the files. So wait for FlowJo to finish calculating, preferably before you do this. Also, as a good practice, save your workspace, which I didn't do before exporting, because sometimes it crashes. All right, so let's click export. You'll get this. It tells you it's writing the files. And then when it's complete, nothing happens. So just close. You can click Browse Destination to see that the files have all exported correctly. And it's important to do this as a check. You should see that files from similar conditions have similar event numbers, or in this case, data file sizes. So all of the blood files are 40 megabytes. The pairs patch is around 20, and the spleen is around 2025. If you see a lot of data files coming out that are 4 kilobytes to 8 kilobytes, that may mean that it has skipped over those and only created a placeholder file without actually putting any data into it. When you quit to R, you will see that these then have 0 events and it can't analyze them. If you see that for a lot of files and you go back into FlowJo and you see that there should be cells in that gate, then you want to export again and just overwrite those files. All right, so let's close this, hit cancel here. And now I'm going to show you how to add suffixes and prefixes to sort your files. So the first thing we want to do is create a group. So we go to the FlowJo menu, we create group. Now for this, I'm going to create a group that says that my blood and my other tissues are in different groups. So we're going to say blood, create group, and now we have a group here, blood. Right now, there's nothing in that group. We can also create a group that says not blood. And let's leave out the hyphen for R and say create group. Now, what's nice is that we can actually name these. We can make these groups a bit smart in FlowJo. So if you have groups, if you're worried about, say, batch effects with human samples, you can say edit group properties, and you can, under keywords here, choose date. So down here, if you type in file, these are the $, you get access to parameters that are written in the FCS file. So for instance, date equals, and then choose In my case, all of these files were acquired the same day, but if you had acquired files on different days and were worried about batch effects, you could use this to segregate based on the date the files were acquired, for instance. We can also use this to, say, sort the files by file name. So file name contains blood. And then we say apply changes. And now all of my 8 files that have the word blood in the file name are in the blood group automatically. I don't have to put them in there one at a time. Now I can close this. For the not blood group, I can do the same thing. And say that that contains spleen. And I can say more choices, or file contains pairs patch. And if I apply changes to that, you can see that I didn't get that correct right because I only have 8 files in there. So I must have named something incorrectly. So we can go to edit, collapse all, and see what our files name. So they're not named pairs patch, they're abbreviated PP. So of course they don't show up. So if I edit the group again, and instead of pair, I say PP and apply the changes, now my extra files show up there. Now the advantage of this is that I can then apply the same gating structure that I have here to those groups. I could also slightly modify the gates if needed for a particular tissue. I don't recommend this in general, but if you have a tissue where you have biological differences, for instance, the lung, where Singlet F can be expressed on alveolar macrophages as well as eosinophils, you might need to use a different gating structure than in the spleen, where Singlet F is almost exclusively on eosinophils. In this case, where the pairs patches have a different expression of CD3 due to a high prevalence of gamma delta T cells than, say, the blood or the spleen, you might consider adjusting the CD3 gate if you saw that there were problems caused by this downstream in the analysis. This would also allow you to adjust for differences in compensation between different days and so on. What we want to do now is export our CD3 cells. And because we have groups, we can do this one group at a time, and we can apply tags to the cells just in this group. So for instance, our not blood, the files in here are called PP or spleen. So right now we have no one label that we can apply to them in R to sort all of these files together. We can add that now by going here, export concatenate group, select all compensator parameters. For the destination, we're going to select the T cells now because we're working with the T cells. We're going to include all events, all compensator parameters, and we're going to say not blood as our group name. And so now you see that this will add a prefix called not blood at the beginning of each of these files. And it's going to be 16 files because there's 16 files in the group. We're going to export this, browse destination, and we can see that now the pairs patches and spleen have this sample tag at the beginning. We can then do the same for the blood, and in this case, we'll just keep it called blood. We don't need to add anything because it already has the label blood in it. Now, one thing to consider with these files is that the naming structure, you need to think about when you are producing them. So if I want to put this modifier prefix at the beginning, I now have a string here with something in the middle between the condition not blood and my next variable that I might be interested in. So I have 3 variables in a row. One is not blood, one is pairs patch, and the third is areg knockout. So if I want to analyze by two variables in a string, I need to either remove one of these, the middle one, in order to do areg knockout and not blood, or generate a different naming structure on the flow cytometer. So we will get to that later. All right, so now we have exported our data files, which just contain CD3 T cells or just contain viable cells. We want to verify that this has worked correctly. So we'll make a new workspace, and then we can drag and drop these files in. And you can see that each of these files, for one, contains cells, which is good. If we open one of these, we can now see that the forward scatter, side scatter is restricted to that area we had before, and there's still singlets. And if we look at CD19 for our B cell marker and CD3 for our T cell marker, you can see that we now have only CD3 T cells. You can also see that our data have now been transformed to be only 10 to the 5, which is something we don't want. So ideally, we need to actually go through and correct this, because otherwise these data are going to get cut off in R, and everything on the axis will read as the same. So unfortunately, you may need to do this for each individual parameter. And some of them it has kept for some reason, and others no, and I don't know why this is. This will probably be different on each machine. You can use this to modify the range of values that you are working with. So for instance, if you have a low expression marker like CD103, you can restrict the data just to 10 to the 5, which may be beneficial for your tSNE separation for that marker. That's a bit more advanced though. In theory, if you do this once, it's supposed to save it. Right, and then we scroll through to check that those parameters have been saved to all of the samples. And it looks like they have. All right, so we can close this. We can also close our original workspace that we were working in. And let's see if I've forgotten anything. Nope. So that is it for this section. And now we will move on to working in our